Welcome to another engaging exploration of genetics. In today's lesson, we will delve into the concept of dihybrid crosses. A cross that involves two pairs of genes or alleles is called dihybrid cross. An easy way to do the dihybrid cross is through the use of Punnett square. For example, let us consider a pea plant that is heterozygous for round yellow seeds crossed with one that is homozygous for round green seeds. What would be the phenotypic ratios of the resulting offspring? To address this question, we will proceed through the following 5 steps, beginning with step 1. Determine the parental genotypes. This information is inferred from the problem statement. The terms heterozygous and homozygous provide crucial insights. From the problem, we understand that the genotype symbol capital R stands for round seeds, small r for wrinkled seeds, capital Y for yellow, and small y for green. The pea plant with heterozygous round yellow seeds has a genotype of capital R, capital Y, small y, indicating heterozygosity for both traits. This means each trait comprises one dominant and one recessive allele. Dominant alleles are denoted by uppercase letters, whereas recessive alleles are represented by lowercase letters. The plant with homozygous round green seeds has a genotype of capital R, small y. The term homozygous indicates that each trait consists of identical alleles. In this instance, round is the dominant trait, suggested by capital R, while green is recessive, indicated by small y. Thus, both alleles for the round trait are dominant and those for the green trait are recessive. Next, we move to determining the gametes produced by each parent. For the first parent, with a genotype of capital R, small r, and capital Y, small y, 4 possible gametes can be formed. These are capital R, capital Y, capital R, small r, capital Y, and small r, small y. This diversity arises from combining all alleles for each trait, with each allele for the other trait. To visualize this more clearly, consider drawing lines between the alleles to see how each gamete is derived. For the second parent, which has the genotype of capital R, small y, only one type of gamete is produced. That is capital R, small y. It is important to note that we list this gamete only once, regardless of whether it occurs multiple times due to the parent's homozygosity in both traits. This approach simplifies the representation of potential gametes for subsequent steps in genetic analysis. Now let's proceed to organize these gametes in a Punnett square. It doesn't matter whether you position the gametes of the first parent at the top or on the side of the square. The end results will be the same. We will place the gametes of each parent along the top and the sides of each cell in the square. Given that the first parent produces 4 distinct gametes and the second parent produces only one, we will arrange them accordingly. Next, we begin filling in the boxes of the Punnett square. When doing so, it is crucial to write the dominant allele before the recessive allele for each genetic combination. This method ensures clarity in understanding the potential dominance of the traits in the offspring. Follow the diagram below as a guide to accurately complete the Punnett square. Finally, we analyze the phenotypes derived from the genotypes in the Punnett square. Specifically, we observe two genotypes, capital Y, and capital R, capital Y, small y, that correspond to round yellow seeds. Additionally, the genotypes capital R, small y, and capital R, small y correspond to round green seeds. Thus, the phenotypic ratio of the offspring from this cross is 50% round yellow and 50% round green. Given that there are four distinct genotypes, each represented in the offspring, this implies that each genotype constitutes 25% of the total offspring. This analysis provides a clear view of how the parental genotypes influence the phenotypic outcomes in their progeny, with equal representation of round yellow and round green seeds among the offspring. Let's analyze the following genetic cross in pea plants. Axial flower possession A is dominant to terminal flower possession a, and tall T is dominant over short t. A heterozygous axial tall pea plant is crossed with a terminal short pea plant. From the designation of heterozygous, it is understood that the alleles for both traits in the first parent must be composed of one dominant and one recessive allele. This makes the genotype of the first parent A, capital T, small t. For the second parent, the traits terminal and short refer to the recessive characteristics. Although the problem does not explicitly use the term homozygous, that inference that terminal and short are recessive traits and can only be expressed phenotypically when homozygous suggests that the genotype of the second parent is small t. This understanding relies on knowing that recessive traits like these are only phenotypically apparent when both alleles are recessive. This setup prepares us to explore the potential genetic combinations that might result from this cross. To resolve the genetic question, we first determine the possible gametes produced by each parent based on their genotypes. For parent 1 with the genotype A, capital T, small t, there are four possible combinations of gametes due to independent assortment, which are A, capital T, capital A, small a, capital T, and small t. These array results from each allele for one trait being paired with each allele for the other trait. For parent 2 with a genotype small t, there is only one possible gamete, and since both traits are homozygous recessive. As a result, all gametes from this parent will be identical, containing the recessive alleles for both traits. This understanding of gamete formation is crucial for the next step, which involves predicting the offspring's genotypes by combining these gametes in a Punnett square. This approach will illustrate the distribution of genetic traits among the progeny. To complete the Punnett square, write the alleles on the box by copying the gamete from parent 1 and parent 2. In writing, don't forget to write first the capital letter followed by the small letter of the same trait. Question 4. What is the phenotypic ratio of the offspring? Can you guess? At its core, a dihybrid cross provides a method to forecast the inheritance patterns of two distinct traits from parents to their offspring. This process offers a compelling glimpse into a fundamental principles of genetics and the mechanisms by which diversity emerges in nature. It's important to appreciate that the true elegance of genetics lies in the variety of it generates. showcasing the intricate and dynamic nature of biological inheritance.